All right, so let me go through this stuff then. So first of all, it says models or pictures. Models or pictures, be able to identify the structures listed. Now, just remember for the practical on Tuesday, I could use a lot of the same models that we have in class. I do reserve the right to use pictures that are similar to it. That goes for any of the labs, labs 1, labs 2, or labs 3. So my only point is don't get too locked into any one model. Make sure you look at lots of images, either in your textbook or online or whatever, just so that you don't like freak out and be like, wait, that's not the model I studied. You know, like, so just make sure you look at lots of different things. All the pictures and models, they're all pretty much similar, but you know, just glance at them to make sure you're comfortable with that. Anyway, so I'll go through this. I'll go through the skin models that we have. I'll look for these things here in a second. Our models don't show enough detail to show you the individual cells. So like the keratinocytes, the melanocytes. So I'd have to use like a picture similar to what was in your textbook or in the PowerPoint to ask you about the individual epithelial, epidermal cells, and I'll show you that picture here in a second when I do the review. These two dumb things, so this was not my decision. This was I don't know who in the A&P department chose this, but these are two things that you're going to learn more about later. They're not really even in the skin. Muscle spindles and Golgi tendon organs are something that are inside your muscles that help you basically sense like how contracted a muscle is or whether a body part is stretched or not stretched. But nonetheless, they want you to be able to identify these things. We don't have any models of those either, but I do have some pictures, and I'll show you those pictures and help you out with that. Then we'll talk about the microscope slides. I'll show you lots of examples of microscope slides. You know, when you look at a microscope slide, you should be able to tell me whether it's thick or thin skin, and then you should be able to identify all those different structures that are listed there. Next, you should tell the difference between thick and thin skin in terms of distribution. That just means like location, where do you find it? And then epidermal strata, that means like how many layers are there to the epidermis and what are the names of those layers. I'll go through that with you as well. And then finally, they also want you to tell the difference between Pacinian and Meissner's corpuscles in terms of what they look like, where they're located in the body, and then what type of senses they're in charge of. So I'll go through all that stuff. So let me go and do these models here first. Whoop, sorry. I know. Just breaking things up here. All right, so here's one of the skin models that we had. So just to get your bearings, everything from here on up would be like the epidermis. And then everything from here to here would be the dermis. And then down here where you see like the fat and stuff, that would be the subcutaneous layer, right? If you look at the epidermis, and by the way, I should just tell you, this is a artist's model that was created. On this one model, there are things that you would see only in thick skin, and there are things that you'd see only in thin skin, but they were combined just so it could show you everything on this model, so I just wanted to make you aware of that. Anyway, if you go up here to the epidermis, and you look at it more closely, this outermost layer that you see here, that's called the stratum corneum. And by the way, on the practical next week, I mean, if you want to be super lazy and not write the word stratum in, stratum is just a fancy word that means layer. So if you just write corneum, I'd probably mark it right, okay? Just below it, it looks kind of yellow. It's a little hard to see, but just below it, that would be the stratum lucidum, okay? That would be a layer that you would only find in thick skin. And then just below that, in kind of a darker blue, or on the microscope slides, it usually looks darker purple. That would be the stratum granulosum. It's usually a pretty easy layer to see. This whole area in here is going to be the stratum spinosum until you get to this very last cell layer that goes here and then up and down. That's the stratum basale that you see there. All right. So those are the layers that you have in the epidermis. And then if you look in the dermis, there's basically two regions, okay? Where you see these little hills up here at the top, where you can actually see them better over here, the area where you have the little hills, that's called the papillary region of the dermis, or you call those little hills dermal papillae, okay? So that's this region. And then The bigger part of the dermis, though, from like here on down, that's called the reticular region of the dermis. And that's where you would find that dense, irregular connective tissue that kind of has collagen fibers that run in all different directions and all that stuff. Okay. Other stuff that you can see on these models, these guys here, of course, are the hair follicles. So here's like an intact hair follicle, and then here's one that's been cut so that you can see inside of it. These little guys that are stuck to the hair follicle here and here, or the cut version, which is over here and over here, those are the oil glands, okay? Or you can say sebaceous glands. I don't really care. You can say oil, you can say sebaceous, but that's what those guys are there. Versus this guy shown here in white, with this duct going all the way up here, that's a sweat gland, okay? Now, I'll show you it on the other model when I get there, but basically sweat glands are long tubes that when you come to the end of them, where the secretory part is, they just kind of twist and turn and twist and turn. So if you end up cutting one of these things, and again, the other model shows it a little bit better, it will look like sort of weird oval-shaped or circular-shaped structures that are kind of clustered together. When you look at microscope slides, you got to be kind of lucky to see the whole sweat gland. Most of the time, you'll see one or the other. You'll either see the secretory part down here and you won't see the duct part, or vice versa, you'll see the duct part but you won't see the secretory part. It's only if you cut it in the perfect plane that you'll get to see like the whole thing. Okay. Sensor receptors that you can see on this model, a couple of ones. So in the dermal papillae, again where the hills are, some of those hills are going to have Meissner's corpuscles, which you see here, there, and then over there is one that's supposed to be uncut. Not every dermal papillae will have a Meissner's, but when you find a Meissner's, it'll always be inside of a dermal papillae. Okay. Another type of sensory receptor that you see much deeper are the Pacinian corpuscles, which you see down here. They're the ones that kind of look like a cut onion. They're always going to be deeper down. They're going to be like in the deep dermis, or sometimes they're even in the subcutaneous layer. Okay? So that's the Pacinian corpuscles. These guys do go by other names, so sometimes Pacinian corpuscles, they call them lamellated or lamellar corpuscles. And these one, the Meissner's, they sometimes call corpuscles of touch, or they call them tactile corpuscles. You're welcome to use any of those names and I'll still mark it right, okay? If you've looked at your textbook, you may notice some different names than what I have in the lab guide that probably mean the same things. If there's ever any confusion, just shoot me an email and be like, does this mean this? And I'll be like, yeah, it probably means the same thing, okay? They just kind of flip around, you know, that's what makes A&P fun, is having multiple names for one thing, all right? Oh, and then one more type of sensory receptor, just to mention it. This guy here that's going in yellow, that you see going up here, this is what's called a free nerve ending. It basically doesn't have any kind of special thing attached to it. It's basically just a bare nerve, but they call that a free nerve ending.
Do you have to write it as free nerve ending or nerve ending cut it?
Yeah, nerve ending's probably okay. I'd probably accept nerve ending. Yeah. Why, is it too much to ask with the word free before it?
No, what my liability.
Fair enough. All right. So that's one of the models. Let me show you the other model, which is kind of bad shape. It's kind of cracked and stuff, but it's got a few things on it maybe that aren't on the other model. So there we go. It took me a while to figure this out.
You can see why it's unfair.
Yeah, that's right. Yeah, you see why it comes at. I used to try to fit this underneath the camera, just putting it up here, and then I realized how dumb that was. I just couldn't do it. So, all right. Okay, so this is just another skin model. Everything from here on up would be epidermis. Everything from here to here would be dermis, and then down here where you see all this fat is basically the subcutaneous layer. With the epidermis, same type of deal. So this would be the stratum corneum. That's the layer, by the way, that's the layer that you kind of want to focus in on when you're trying to tell the difference between thick and thin skin. The stratum corneum will always be really thick in thick skin, versus in thin skin, it'll be really thin. You won't see much of it. Next, you can barely see it, but kind of shown here in white, that would be the stratum lucidum. That's a layer that you only find in thick skin. Honestly, it can be hard to see it in thick skin sometimes, but it should be there, whereas you won't have it in thin skin. The next layer, which is usually easy to see because it's like dark purple, that's the stratum granulosum. Then this whole area in here is the stratum spinosum, and then this little layer that sits right next to the dermis, that would be the stratum basale. Okay. These little hills that you see, those are the dermal papillae, so that's the papillary region of the dermis. These things that you see here in red and blue, you don't actually need to know it for the lab practical, but that was what that question was that I gave you. Those are the capillary loops that I was asking about before during lecture, but you don't have to worry about that for lab purposes. So that's the papillary region. Then the rest of the part of the dermis would be called the reticular region, right? Once again, these are some hair follicles that you see there. These would be some oil glands here and there, and then over here, and then over there. This is almost identical to what the oil glands look like under the microscope. It's actually a pretty good representation of what oil glands look like under the microscope. Also on this slide, I kind of like the fact that it's got several sweat glands. So here's a sweat gland, there's another sweat gland, and then over here is one that's actually been cut. And I like this one because it kind of illustrates, you know, if you have a long tube that twists and turns, and then you were to cut that, what's that going to look like? It's going to look like a bunch of weird sort of oval-shaped or circular-shaped structures. That's what this is going to look like when it gets cut, right? And you have to sort of be able to see that. Now, only if you get really lucky when you make the microscope slide, though, will you actually be able to catch the entire duct going up to the surface, right? These things are very, very small, so they got to be cut. You know, you got to get kind of lucky when you cut them and hope. Just like you kind of get lucky when you cut the hair follicle, you may not see it to go all the way up to the surface, right? Other stuff to see on here. This would be an example of a Meissner's corpuscle. It's inside of a dermal papillae, so that's how you know it's a Meissner. This big guy down here that's deeper, that would be a Pacinian corpuscle that's sitting down here. Now, one thing that this model has that the other one doesn't have is that it's a third type of sensory receptor. This guy over here that almost looks like the letter T, that's a Ruffini corpuscle, okay? They all do different sensations. We don't really have to get too far into the details. I will talk about the sensations for the Meissners and the Pacinian later, but just make sure you can identify like that's a Ruffini when you see it like that. All right, I think that's all I want to say about this model. So let me go on to the microscope slides. Actually, let me show you the pictures of the epidermal cells, and let me show you the muscle spindle and the Golgi tendon organ. Whoops, didn't mean to do that. Okay, so here are the epidermal cells. We just don't have a model that gives this level of detail, so that's why I got to use a picture like this one. Will I use this exact picture? Maybe, or maybe not. I'll use something similar to it, so just look online, you'll find other pictures too. But remember, if you're just playing the odds, most of these cells that are scattered around here, the ones down here, even the thin ones that are at the top, most of those guys are keratinocytes, right? They make up the vast majority. Sitting somewhere in the middle of the stratum spinosum, you'll usually find a Langerhans cell. Langerhans cells go by a couple of different names. I think Langerhans cell is probably the easiest way to say it, but if you want to be an A&P snob, you could call it an epidermal dendritic cell or an intraepidermal macrophage, right? Whatever. In the stratum basale, that's where you want to look around for melanocytes. They kind of look sort of weird, octopus shaped. They're going to be sitting down there. If the artist does what this artist does, they may actually draw little black dots to represent like melanin that's being produced, right? And then this guy over here, that would be one of those Merkel cells. It's also known as the tactile epithelial cell. It's a type of sensory receptor cell. And you can always tell a Merkel cell because sitting right next to it will be the end of a neuron, which you can see there. Okay. Now this is stupid, but I have to talk about it anyway. So this is that thing called a muscle spindle and a Golgi tendon organ. The muscle spindle is the guy that's inside of the muscle. So this is the muscle spindle. And then this thing here is the Golgi tendon organ. These are sensory receptors that detect how stretched a muscle is and how much contraction is built up in a muscle. You don't really have to worry about that now. We'll talk more about them later, but you should know this picture. And if I point to this thing, or I circle this whole thing, you would call that a muscle spindle. If I circle this thing, you would call that a Golgi tendon organ. And then also they want you to know, this is so dumb, I should just delete this. No, I have enough power now. Yeah, you can't boss me around. I've been here for 12 years. Who are those guys? Yeah, it's making my blood boil. I'm getting infuriated just talking about this. Anyway, those are called intrafusal muscle fibers. They're the ones that actually make up the muscle spindle. Intrafusal fibers. The rest of the regular muscle cells that you see that are not part of the muscle spindle, they call those extrafusal muscle fibers.
So there's going to be a question on that on the practical?
There could. It's so dumb,
When you
right?
say there could, there's like...
There is.
By could, I mean is. Yeah, no. I mean, to be honest with you, Valentina, I don't even remember if I put this on there. I might have been so grumpy that I just left it off. I don't know. But you know what? That's why I make the big bucks, because I have to give you news that no one else will. I don't know. All right. All right, so let's talk about microscope slides. So first off, is this thick or thin skin? What's your guess?
I think it's thick.
It is thick, yeah. So I was just going to interrupt before you had a chance to second guess yourself. It's thick. This up here is all stratum corneum, okay? That's really thick. Just trust me on that. I'll show you examples of thin skin in a second, but the stratum corneum would not be that thick if it were thin skin, number one. Number two, another tip-off that might lead you in the direction to think this is thick skin is what don't you see?
Hair follicles.
You don't see any hair follicles, right? So that's probably another good tip-off. Now, is it possible that you just happen to catch a part of your body where there's not a lot of hair? Yeah, that's possible. It could still be, but especially if you look at the stratum corneum, that should kind of guide you in the direction. Another tip-off too is, what's this white layer that you see here? That's the lucidum. That's that layer that you only see in thick skin. So that should be another tip-off. Then as you go below it, where it's kind of darker purple here, that's the stratum granulosum right there. Again, it's sort of subtle, but if you kind of squint, you can see how it's a little bit darker purple in that area. Then in here, that would be the stratum spinosum. And then this last layer that sits right next to the dermis, that would be the stratum basale. Where these hills are, those would be the dermal papillae, or if you want to call it the papillary region, either one's fine. And then down in here, this would all be reticular region. And that's where you'd find the dense, irregular connective tissue. Is this thick or thin? It's also thick, all right? Again, everything from here on up is stratum corneum, okay? That's very thick, all right? So you know that's thick skin. This thing, now it's shown kind of in a light blue. That would be the stratum lucidum coming through there in the light blue. And then the darker purple that's right underneath it would be the stratum granulosum, okay? Then you get into the stratum spinosum, and then the layer that would be right against the dermis would be the stratum basale. These would be the dermal papillae, and then the rest of this would be the reticular region. Okay. Which is this one, thick or thin? This is thin. Okay, fine, we got an example of thin. Notice this thing that's kind of looks like it's peeling off here, that's the stratum corneum, okay? There's not much to that, right? That's not nearly as thick as, you know, this big guy, right? So that's a good tip-off right then and there. And that's actually a really good tip-off here because, you know, you don't really see any hair follicles on this slide. So you might be like, wait, you know, is that thick skin? There's no hair follicles. Well, no. Look at this, and you'll see that's pretty darn thin up there. So it is actually thin skin. You know, it's hard to see, but like this kind of darker purple line here, that would be the stratum granulosum. Then in the middle, you'd have like the stratum spinosum, and then down at the bottom here, you'd have the stratum basale. It's really hard in this spot where they took this, for whatever reason, to see any dermal papillae. It's kind of hard to see any hills there. They're sometimes not as well defined as they are in thick skin. That could be another reason. The rest of this area down here is all going to be reticular region of the dermis. Is this thick or thin? This is thin. And finally, again, let's just start, before we dive into the obvious, let's start up here at the top. Notice this kind of light pink or light purple that's on there. That would be the stratum corneum. It's not very thick, so that's a good idea there. But obviously the real tip off is what? What do you see here?
Hair follicles.
Hair follicles. All these things that are coming in at an angle, those are all hair follicles. Okay, and in fact, like here, they actually still have a little bit of a hair there. You can see a little bit of a hair there. Like I tried to tell several people, unless you get lucky, you may not see the entire hair follicle. Like they did a pretty good job on this one. You can actually trace it all the way down. But like this one, you're just kind of seeing the bottom part, right? You're not seeing the rest of it. Or this one and this one. This is all the same hair follicle, but it kind of looks like two separate structures because they didn't cut it in the perfect plane, right? Other stuff you can see on here. So again, up in here would be the dermal papillae. This would be the reticular region of the dermis. Down here where you see a lot of adipose tissue, this would be the subcutaneous layer. And then what are you seeing like here or like there? Those are the oil glands, okay? They're emptying into those hair follicles. Now I have some better magnification, but just based on their kind of position, it'd be a pretty good guess that those are going to be sebaceous glands or oil glands. Now also what you can see here too, and I have much better pictures in a second, you see these little round structures that are kind of down here? That's going to be the secretory part of a sweat gland that's sitting down there, okay? I'll give you much better views of sweat glands in a moment, but I just wanted to show you that. Is this thick or thin? Yeah, it's also thin. You can see a ton of these hair follicles. And here I did this one just mostly because you can see a larger view of the oil glands. So there's some oil glands there. Looks like there's some over there. There's some oil glands here, right? And they're all emptying into those hair follicles. Is this thick or thin? It's still thin, okay? This is a hair follicle over here, and there's a ton of oil glands in this area here. You can see them all hanging out. But another nice thing about this one is you get a good view of some sweat glands. So this would be a sweat gland here. Again, that's the secretory part. If you can envision it, think of a tube that's twisting and turning, and then you cut it. What's it going to look like? It's going to look like a bunch of little circles or ovals, right? So that's the secretory part. Do you see a duct? Well, no, you don't see a duct. That's just because when they cut it, they didn't get lucky enough to catch the duct. You see the secretory part. Same thing over here, even more obvious. That's another sweat gland where you're catching the secretory part of the sweat gland. Okay? You don't have to specify secretory part. You can just, if I just point to if I point to this or I point to that, you can just say sweat gland and be done with it. Okay? This is a really good slide with a lot of detail. So this is obviously thin skin. They're not even bothering to show you the epidermis on this. They're just kind of focused in on the dermis. But you can see this, nice looking hair follicle here. And then these really good view of these oil glands, right? And it looks pretty much identical to what, what you can see on that model. Again, there's more oil gland over there. And then this is also a really good view of some sweat glands. So this cluster here is a sweat gland. This cluster here is a sweat gland. And then this cluster over here is a sweat gland, okay? Unfortunately, as good as it is, they didn't get lucky that you can actually see the duct going all the way to the surface, but nonetheless, you should be able to say that's a sweat gland. This guy, on the other hand, wins the prize, right? They got lucky, and somehow, I don't know how, they paid somebody off, I don't know what they did, or maybe they just spent like all day there trying to cut this just right. You can see the secretory part, and bonus, you're actually seeing the duct go all the way up to the surface, right? So that's a pretty good view of a sweat gland.
Is that thin or thick?
In this particular one, this actually looks to me like it's probably thick, just based on, that's all stratum corneum up there, okay?
What's that red one to the left? Is that not a hair follicle?
It's hard to know what that is. I think it's probably, I think it's probably a little blood vessel that's maybe packed with a bunch of red blood cells, I'm guessing. Okay? It's kind of strange. It's possible that this is thin. It's just kind of weird how thick the corneum looks there, so that's why I was calling it that way. Here are a whole bunch of sweat glands. So every one of these dark things that you see is a sweat gland. And on this particular one, they got lucky so that they actually traced the duct going up, right?
That's big, right?
Well, this you can't even tell. They're not even they're not showing you the epidermis on this, right? I mean, based on the fact that we know there's more sweat glands in thick, it probably is thick. I mean, there's a lot of sweat glands there, but without seeing the epidermis on this, I really wouldn't call it. Yeah. All right, now this, first of all, is this thick or thin skin? It's thick. Look how thick the stratum corneum is. You can barely see a stratum lucidum. If you have a good imagination, you can kind of convince yourself that maybe this is a little bit lighter pink through here. Then you can see the stratum granulosum, okay? And the spinosum, and then the stratum basale. But what this slide is trying to point out to you pretty obviously, because it's got a pointer there, is what? What's that, do you think, supposed to represent? That's a Meissner's corpuscle, all right? Now, in terms of the shape or the structure of Meissner's corpuscles, I don't know who came up with this, but someone said it looked like raw cotton. If you took raw cotton and you pulled the seeds apart. Has anyone here recently grabbed some raw cotton? All right, me neither. But apparently there's little seeds in raw cotton, and if you pull it apart like that, it looks sort of like that. Or if I were to draw it, it'd be kind of like this. You got the fibers of the cotton that are kind of pulled back and forth like that, and then off to the edges, you see like these little black spots. Those would be kind of like the seeds. And then if this whole thing was sort of encapsulated like this, that's sort of what a Meissner's corpuscle looks like. Meissner's, again, you'll only find them inside of dermal papillae. Not every dermal papillae will have one, but when you find one, it'll be inside of a dermal papillae. Okay? Here's a close-up of one that you can kind of see that sort of pulled apart cotton appearance that they're talking about with like the little seeds on the edges and then like the fibers running across.
The V6.
Here is a Meissner's, and then over there is another Meissner's. It's pretty subtle, but you can see, once you kind of trick your eyes, you can kind of be like, okay, I see that. Whereas these other dermal papillae, they don't have a Meissner's inside of it. What's this? That's a Pacinian corpuscle, right? That's the one that looks like this, more of a cut onion, okay? Here are, if you're wondering what 3 Pacinian corpuscles look like, that's what 3 look like next to each other. Now this is actually one that's very similar to the slide you guys had. So this is a Pacinian corpuscle, and it's actually in the pancreas. So the one that Jess saw and some other people saw, this is from the pancreas. It really doesn't matter whether you're looking at the skin or at the pancreas. The Pacinian corpuscles look the same. Why am I showing you ones from a pancreas if we're doing the skin? I think my answer to that is it's just easier to find them in the pancreatic tissue than it is in the skin, and that's why we have those slides. But regardless, they always have that kind of cut onion looking appearance. Okay. Now, the last 2 parts of the lab ask you to make comparisons between thick and thin skin and then between Pacinian and Meissner's. This is a table from your book. You don't have to memorize the whole table, just these 2 areas. So the first question is kind of the distribution, also known as the location. Where do you find thick skin? Palms of your hands or the soles of your feet. Where do you find thin skin? Everywhere else. Okay, that's what they mean by distribution. And then epidermal strata just means how many layers do you have and what's the difference. Remember, with thick skin, you have all 5 layers, including this thing called the stratum lucidum. With thin skin, you basically just have 4 layers. You don't have the stratum lucidum. Okay? And then last, the Meissner versus Pacinian corpuscle comparison. First is, the structure and the location. So again, the structure of a Meissner's corpuscle is looks sort of like cotton, raw cotton that has been pulled apart and has that kind of appearance there. Where's the location? Inside dermal papillae, right? That's where you always find Meissner's. What type of sensations do they do? Well, it says onset of touch and low frequency vibrations. That would be fine if you want to write that if I asked that question on the lab practical. You could also, and you might just want to write this off to the side over here, just write light touch. All right, so because Meissner's are close to the surface, they're one of the first ones to be stimulated when something touches your arm. So if something starts to touch your arm, it's probably either going to be a Meissner's corpuscle or maybe a Merkel cell that picks it up just because they're close to the surface. So that's what I mean by light touch. Versus the Pacinian corpuscle. So the Pacinian corpuscle, first of all, what do they look like? They look like a cut onion. Where do you find them? They're going to be deeper. They're going to be either in the deep dermis or maybe in the subcutaneous layer. And they might even also be in some of your internal organs like the pancreas. I mentioned that before. What kind of sensation do they do? Now, up here they wrote high frequency vibrations. I would just write off to the side, and then you're welcome to write this if you want, but I think it's easy to remember deep pressure. So light touch for these guys, deeper pressure for these guys. And that's because, again, of their location. They're located deeper down. So in order to stimulate those guys, something has to press on your skin more strongly just to even stimulate it. So more of a deeper pressure type sensation. Okay? All right, I think that's all there is. Any questions about any of the stuff we went through with the skin lab or any of the previous labs or anything like that? All right, well, have a nice weekend. If you think of a question,